This is a brief video introducing some aspects of the immune response that include cells and cytokines, how they trigger, how they are specialized, mature, and maintain an immune response. The first step in the immune system response to danger signals is an antigen presentation. This antigen presenting cell, depicted here as a dendritic cell, travels in the bloodstream carrying pieces of the antigen that is perceived as danger signal, and they become in contact within the lymphoid organ with the naive T cell. This naive CD8 positive T cell will identify the antigen through the T cell receptor depicted in green, and the signal from the dendritic cell or the antigen presenting cell that tells the T cell this is a true presentation of a danger signal is the chemokines that are co-stimulatory chemokines depicted here as the XCL1 and corresponding XCR1, the receptor to that ligand, on the T cell. This is what really tells the T cell that what they're identifying via the T cell receptor here, depicted in green, is real. They should respond to it. And the first response is basically by short-lived effector cells. These are mostly going to die very quickly, but they are the ones that are going to be like the first line of defense in this recruitment. A subset of cells are actually the ones that we know are called memory precursor effector cells. There's less of these, but they will be remaining in the lymphoid system in case the memory is required for future contact with the antigen. But so far, the dendritic cell has come from the infected site to the lymphoid organ and just sort of given the alarm to the T cell that now has been primed to respond. In a next step, there's a preparation of these T cells by expanding these clones that now have recognized the antigen. They are still in the immune system, but they're starting to tell each other they have to be ready to fight the antigen. And they also have to differentiate. The differentiation takes different steps. The interleukin-2 signaling will actually stimulate the short-lived effector cells for differentiating above the memory cells. In a first step, they're telling them to be ready to the attack very quickly. On the other hand, in the environment, depending on the type of antigen, the type of antigen-presenting cell, and also the type of co-stimulatory signal, we have now a differentiation that if driven by IL-12 or interleukin-12, it will produce a phenotype on the T cell which is called Th1 phenotype, characterized by the secretion of interferon gamma and TNF-alpha. On the other hand, if it's driven by IL-21 or interleukin-21, it will trigger basically an IL-17 or Th17 response. IL-27 I will mention later because this is a very important pathway that will trigger a control signal by basically secreting interleukin-10. But I'll talk about this at the end of this presentation. The important thing is not just the differentiation, but also the activation of these T cells. And the way that they become activated is they also will actually express in the surface the migration chemokine. These migration chemokines are going to be critical for them to now go to the site of infection, because so far everything has happened within the lymphoid system. There was a signal of danger that was presented by an antigen-presenting cell. This is like in a fire. The signal has come to the fire station, but the Dalmatians have to actually drive the fire trucks to the site of the fire to deal with it. So now we have the chemokines that are expressed that tells us those T cells have been now activated, they have been differentiated, and they expanded, and they are ready in circulation they travel, and what they do with that chemokine expression is that they actually will identify the corresponding ligands of the very specific cells that are infected. And this all has to do with the system that presented the antigen to the T cell. There's communication between the infected cells in situ to the antigen presenting cells that take it to the CD8 T cells, and the CD8 T cells will express the appropriate chemokine or CXCR3 that will identify the cell that is infected, and that's what allows them to migrate. They bind now these T cells, and they actually are pulled into the site of the infection, and that's how they produce the killing of the danger signal. This killing by the activated T cells, now that have been differentiated and expanded and they are in situ, they could be CD4 dependent or CD4 independent. The CD4 T cell dependent pathway will present the antigen and activate the antigen for the CD8 primary response to deal with them. But there are other antigens or other danger signals that are independent of the CD4 signaling. And in this case, they will actually just deal with them directly with the CD8 T cells. In order to explain this a little bit more, I want to refer to the MHC class 1 and MHC class 2. So this is the way that the antigens will be processed. Depending on the antigen presenting cell will actually be the one that is infected by this virus or this danger signal. So this danger signal goes into a cell, and if this particular antigen is presented by the MHC class 1, the MHC class 1 interacts directly with the CD8 cytotoxic T cell, and it will produce a killing of the infected cell. And again, the presentation via the MHC class 1 depends on the antigen and on the cell that is actually digesting that antigen. And it directly causes the killing of that infected cell. On the other hand, other type of antigens are processed and presented by the MHC class 2. MHC class 2 is a bit more complex in the way that it actually will engage now the CD4 T cells. And these CD4 T cells that are engaged by the MHC class 2 with an antigen, what they do is they secrete cytokines. These cytokines will activate the macrophages that have the virus or the pathogen inside, and they will activate it to destroy it. So it's a different mechanisms. CD4 T cells are not killer cells, but they stimulate the other cells to kill it. CD4 T cells can also be engaged via the MHC2 by B cells. So when this particular antigen is seen by the B cell and processed by the B cell and presented via the MHC class 2, they will now engage the T cell. And likewise, the same as before, these CD4 T cells that have been presented an antigen via the MHC class 2 will produce cytokines that stimulate, in this case, the B cells into proliferation, maturation, and production of plasma cells that will now generate and secrete antibodies that are specific to that antigen. In the MHC class 2 response, you can have a combination of the two. You can have the direct activation of macrophages together with a B cell response. It's what we can have a cellular response and a humoral response by presence of antibodies. But again, depending on the type of danger signal and depending on the type of activation and presentation to the T cell, we have CD4 dependent responses or CD4 independent responses, where the major killing cell is the activated CD8 T cell that was migrated and that was actually engaged original in the lymphoid organ. Now, a very important thing of the immune system is an internal clock. This internal clock is a very interesting pathway where, interestingly, is precisely these CD8 active T cells the major producers of a single cytokine, which is called interleukin-10. Interleukin-10 is basically produced in the peripheral system, in the peripheral tissues, by the CD8 T cells, the same ones that produced inflammation. But it is very important to say that publications have more and more identifiers for where are the genes for IL-10 and which cells can actually synthesize IL-10. And I don't need to read you the complete list because there's growing and growing, but basically they can be Th1, Th2, Th17s, Treg cells, they could be B cells, NK cells, macrophages, dendritic cells. What this is really telling us, the message behind this multitude of different kinds of cells having this single gene and the ability to produce this single cytokine is how important this cytokine is for us to be able to live. While it's crucial to have an immune system that is intact and that can deal with danger, it is as important to have a mechanism that controls that immune system and brings it back to inactivity. So interleukin-10 is a critical cytokine that will control the response of the immune system. And there's still a lot more to know about how IL-10 works. But basically what we do know is the cytotoxic lymphocyte will actually need to have the CD4 T cell IL-2 mediated stimulation. If you remember at the beginning of this talk, IL-2 was one of the cytokines that produced the short-lived effector cells. But in this case is now in the periphery producing a stimulation to actually start synthesizing the interleukin-10. In addition, via the T cell receptor that identifies the antigen that is presented by the infected cell, we also have the activation of the internal pathway for the synthesis of interleukin-10. And finally, as I mentioned before, interleukin-27. Interleukin-27 is critical for the control of this mechanism, and it can be secreted by innate cells, and all of these pathways are requirements for the cytotoxic lymphocyte to start producing interleukin-10 and dampen inflammation and tissue damage. I hope this was useful and that this is helpful for you to understand the complexity of responses of the immune system. Thank you.